Hi, everybody. Thank you for joining. My name is Mary Madden, and I'm the Assistant Director of Programs and Grants at C2ST. Welcome to today's program. This program is presented to you by the Chicago Council on Science and Technology in partnership with the Carl R. Woese Institute for Genomic Biology, or IGB, and it's sponsored by the Kleinman Center for Genomics in Business and Society. The Carl R. Woese Institute for Genomic Biology is where science meets society, an interdisciplinary institute dedicated to transformative research in technology and life sciences using team-based strategies to tackle grand societal challenges. The program is sponsored by the Don Catherine Kleinman Center for Genomics in Business and Society. The Kleinman Center serves as a platform for enhancing and accelerating the broader impact of the Carl R. Woese IGB's research and innovation, building on IGB's innovative genomics research by enhancing its global influence. The center helps IGB provide unique opportunities for economic development, public engagement, and social impact. For those of you that are new to the Chicago Council on Science and Technology, C2ST is a 13-year-old not-for-profit organization that seeks to enhance the public's understanding and appreciation of science and technology and their impact on society. C2ST hosts around 30 free or low-cost programs throughout the year on any and all science and technology topics. We're still focusing on virtual programming now, but stay tuned for a return to in-person programming once it's safe. Please visit c2st.org to learn how you can donate and get information about C2ST's upcoming virtual programs. Remember to sign up for our mailing list so that you can get reminders about these programs as well. And today, please stay tuned after the program if you're interested in learning about or joining C2ST's auxiliary board, where you can connect with young professionals who generate and shape our public discourse about science in Chicago and beyond. C2ST's Aux Board guides programming, outreach, fundraising, and a whole slew of other areas focused on communicating and partaking in science. So just a couple of housekeeping items before we get started today. Please type c2st.cnf.io into your web browser and click today's session to ask questions throughout the talk and evaluate the program at the end. You'll also be able to ask questions from wherever you're watching. Our staff will be checking C2ST TV YouTube Live as well as Facebook Live for your questions. Again, to ask those questions directly, you're going to go to c2st.cnf.io. And now I'd like to introduce our speaker for today. Dr. Thomas Kehl-Fie is an assistant professor of microbiology and affiliate member of the Carl R. Woese Institute for Genomic Biology at the University of Illinois at Urbana-Champaign. Dr. Kehl-Fie's investigations have significantly expanded the scope of nutrients known to be withheld by the host during infection. He also developed a series of unique tools derived from host proteins responsible for restricting those nutrients, which enable the impact that nutritional immunity has on pathogens to be studied both in culture and during infection. And without further ado, let's welcome Thomas.
Thank you, Sasha. Thank you, Mary, for that introduction, and thank you, everyone in the audience, for coming today and listening to this talk. I appreciate the opportunity to talk to the C2ST group. Before I get to the meat of my seminar today, I'd really like to start by acknowledging all of the people who have done the bulk of the work, and that's the people who work every day in my research lab. It's a team effort with students, postdocs, and staff scientists doing a lot of the work, as well as collaborations with people in the mining microbial genomes theme in the Carl R. Woese Institute for Genomic Biology, as well as collaborators around the world. The work that I'm going to talk to you today about was primarily conducted by a really talented graduate student who just completed his doctoral studies, Kyle Grimm, and is now actually working in the Chicago area, and a postdoc, Jacqueline Moray, who's recently returned to Australia and once again is working in industry, developing new therapeutics to treat microbial infections. Before I get too far into my seminar, I just want to mention that, my son is at home with me, and he's really excited about kindergarten. So if you hear anything in the background, that is him. The other thing I'd like to mention is that I'm fairly certain that I don't look like this. Now, this is a bottle of bacterial growth media, and this is how we typically study bacteria. Now, if you ask my son, he'd tell you, Well, Dad, you actually kind of look like that. There's your head and there's your body. Now, when he tells me this, I take solace in the fact that all of the pathogens, all of the microbes that live on my body and try to make me sick disagree with him. This is a virtual buffet for bacteria. It's nutrient rich, it has everything they need, and is really a happy place for them to grow. Our bodies, however, are a really nutrient poor environment that's always trying to kill them. An example of this can be seen right here. This is the liver from a mouse that's been infected with a bacteria. And we can see healthy tissue here, and we can see the site of infection right there. Using a technique that allows us to take a picture of the metal distribution within a tissue, we see that the healthy tissue is replete in metals such as zinc and manganese, but the sites of infection are nutrient poor or metal poor. Now you're maybe thinking, well, okay, so metals aren't available. Why do the pathogens, why do the bacteria that are living on and trying to make me sick care about this? And the answer to that is that metals are absolutely essential for all forms of life. Metalloproteins, or the structures that allow our bodies to do things, about 30% of all of those proteins use a metal cofactor. 50% of all enzymes use a metal cofactor. So without metals, our bodies and pathogens can't make new compounds, they can't generate energy, they can't synthesize new proteins, they can't synthesize DNA, the heritable molecule of life, and they can't resist the stresses that the host immune system uses to attack them. But at the same time, we know that pathogens are capable of causing devastating disease. So what my laboratory is really interested in understanding is, how do pathogens overcome host-imposed metal starvation, or the defense known as nutritional immunity? Now, if we take a step back, and before we do any experiments, we think about this, we can come up with two broad ways that pathogens might overcome host-imposed metal starvation. One, they can work harder. You can think of this as a tug of war. The host is pulling metals away from the bacteria, and so the bacteria start to work harder by making transporters and other things to get metals back from the host. The other way that pathogens might adapt to nutritional immunity or metal starvation is they might work smarter. The host takes away the metals. The bacteria might try to reduce their reliance on these metals. Now, while I'm going to tell you a story about pathogenesis, what I'd like to suggest to you today is that these studies have the potential not only to inform how bacteria make us sick, but how microbes live in the environment writ large and may improve other aspects of our lives. Now, every good story needs a protagonist, and the protagonist that we're going to talk about today is Staphylococcus aureus. Staphylococcus aureus is a devastating pathogen capable of infecting nearly every tissue in the human body. What makes Staph aureus particularly nasty is the emergence of antibiotic resistance. One antibiotic is resistant to in particular is methicillin. Methicillin infections have been diagnosed, or are frequently diagnosed, around 300,000 infections per year in the hospital setting. This causes about 10,000 deaths per year and about $1.7 billion in costs to the United States healthcare system every year. Now, if we want to put the total burden of these infections in a little more understandable context, this is about 6 times the student population of the University of Illinois, or about 6 times the capacity of Soldier Field. So Staph aureus and other pathogens are really a dangerous threat to humanity, and we need new strategies for treating their infections or preventing their infections. Now, so we have our pathogen that we're going to study, but I just told you a moment ago that the medias we use to study pathogens are nutrient rich. They're metal rich. The question then becomes, how do we study nutritional immunity? How do we study metal starvation? To do this, we took a page out of the host's playbook, The host uses proteins to strip metals away from the pathogens or take metals away from the pathogens. One of the important players in this host defense is a protein called Calprotectin. Calprotectin uses two transition metal binding sites, or two metal binding sites, to bind metals extremely tightly, preventing pathogens from acquiring the metals. So what we decided to ask is, can we use Calprotectin as a tool and its intrinsic ability to bind metals to steal metals away from pathogens when grown in culture, allowing us to study this host defense. So what we did is we grew a variety of pathogens, including Staph aureus, in media, and we added Calprotectin, and we just asked, how much did the bacteria grow? I'll frequently show you growth assays where larger bars indicate that the bacteria grew better, while lower bars indicate that the bacteria grew worse. As we can see here, that when we add Calprotectin to all of these bacteria, it's capable of inhibiting growth. And we know that this growth inhibition is occurring because of its ability to bind and sequester metals, telling us or giving us a way now to study nutritional immunity in culture, allowing us to answer those questions that we posed, that question we posed earlier. So if we think about the struggle for metals, we think about what's going on during infection, we can envision that when Staph aureus and other pathogens first come to a site of infection, it's a relatively metal replete environment. However, the host senses that the pathogens are present and it restricts the metals. It takes them away. This in turn leads the bacteria to make transporters that allow it to fight with the host or pull back and try to get those metals. We know from prior work that to obtain manganese, Staph aureus makes two different transporters, one called MNTH and one called MNTABC, and these transporters are critically important for the ability of Staph aureus to cause infection. If you eliminate Staph aureus if you eliminate the MNTABC transporter here, Staph aureus is no longer able to cause disease. And a number of companies now are trying to develop vaccines and other therapies that target this manganese transport system. However, we also know that Staph aureus has to obtain zinc, and we know that Staph aureus is actually fairly good at obtaining zinc during infection. But we don't know how Staph aureus successfully competes with the host for zinc. We don't know what the transporters are that Staph aureus uses. Now, while we don't know what the transporters are that Staph aureus uses, we do know how other bacteria obtain zinc. And one of the systems that bacteria use is called the ADC system. This allows bacteria to move zinc from the outside of the cell to the inside of the cell. So we just wondered, perhaps this ADC system is important for the ability of Staph aureus to resist Calprotectin. Now, Kyle, the graduate student who was working on this project, did a lot of work. And then he did a lot more work. And then he did a lot more work. And then he came back and said, no, the ADC system isn't essential to the ability of Staph aureus to resist Calprotectin and overcome the zinc starvation that it experiences during infection. Now, this left us with a conundrum. We knew that Staph aureus needs zinc. We knew that it has to be able to get zinc, but we didn't know how it was doing this. So we used transcriptional profiling, which just allows us to ask, how does Staph change when you expose it to a stressor? To ask, well, what does Staph turn on when you expose it to Calprotectin, which would suggest, or the things that it turns on, we think would be important for resisting Calprotectin, and these transporters that it must use would be a subset of those genes. In this analysis, we identified another transporter called the CNT system. Now, the CNT system has never been associated with zinc transport, but it we thought might be the system that would be allowing Staph aureus to compete with the host for zinc. Now, metal transporters in bacteria are frequently turned on when the bacteria get hungry. Bacteria have proteins, sensors, if you will, that sense that a bacteria gets hungry, and then when the bacteria is hungry for a specific metal, that protein turns on expression or allows expression of the metal transporter. There's several different regulators. One is called ZIR. It allows bacteria to sense zinc availability, and if they're hungry for zinc, One is called MNTR, which senses manganese availability, and another is called FUR, which senses iron availability. We generated strains of Staph aureus that lack each of these regulators individually, which makes them think they're always hungry. When we look at gene expression, or we look at the expression of ADCA and the CNT system, we see that loss of the zinc uptake regulator increases expression of the ADC system and increases expression of the CNT system, but not when we knock out or eliminate MNTR or FUR, telling us that both of these systems are regulated or turned on when zinc is absent, suggesting a role in zinc uptake. The next thing that we did to ask, well, do you really have a role in zinc uptake, is we went back and took advantage of our ability to mutate Staph aureus or eliminate genes from Staph aureus, and we eliminated the ADC system, we eliminated the CNT system, and we eliminated the ADCA system and the CNT system together, and then just grew the bacteria in the presence of Calprotectin. And so what we can see here is that in low concentrations of Calprotectin, neither the ADC system or the CNT system are essential, but if you eliminate both of them together, the bacteria have a growth defect, meaning that together both of them are necessary, or one of or the other is necessary when the bacteria are just a little bit zinc hungry. But as we increase the concentration of Calprotectin, increasing the hunger or the starvation that the bacteria are experiencing, we see that the CNT mutant, or the strain that lacks this second or new potential zinc transporter, begins to grow worse. And this tells us that Staph aureus has two zinc transporters, but that the CNT system appears to be the one that's more important for resisting host proteins such as calprotectin that restrict metal availability. Now, everything that I've shown you so far occurs in a test tube, in culture. And even though we can add calprotectin and add other factors to that to make that look more like an infection, it can't recapitulate everything that occurs during infection. So what we did is ask, if we take our strains that lack the ADC system, lack the CNT system, or lack both of them, what happens in the context of infection? We can compare that to mice that have been infected with wild-type bacteria, and what we see is that when we eliminate the ADCA system, there's no defect in virulence. When we eliminate the CNT system, we see a reduction in virulence, and when we eliminate both of these zinc transporters, we see a further reduction in bacterial virulence. So this tells us that Staph aureus uses two transporters for manganese and two transporters for zinc. And we also know that the CNT system is necessary and the primary system used by Staph aureus to obtain zinc from the host during infection. Now, the CNT system is unique from the other transporters in that it's encoded here, but immediately upstream and downstream of it, there's additional genes, there's accessory factors. And while we were working on the CNT system, another group described these synthesis genes as producing a small molecule that has a high affinity for metals, a metallophore. And this gene downstream here, labeled MFS or CNTE, as a protein that secretes this metallophore. And so we were wondering now, perhaps the CNT system, the CNT transporter, works in conjunction with these genes here to bring zinc into the cell. So put another way, is Staphylopine, that metallophore made by the CNT loci, or CNT system, a zincophore? So if this is a zincophore, we would have the CNT-KLM accessory genes producing the metallophore. They would secrete it via CNTE. It would bind zinc, and then it would be transported back into the cell by the CNT-ABCDF system, or transporter. Now, if this is the case, both KLM and CNTE, the synthesis and efflux machinery, should be essential for resisting calprotectin-imposed zinc starvation. So we once again took advantage of our ability to mutate Staph aureus and eliminated the synthesis genes and eliminated the efflux genes. And what we saw is that as we exposed them to calprotectin, they grew worse than wild-type bacteria, telling us that production and secretion of Staphylopine, or this zincophore, is a potential zincophore, I should say, is essential for Staph aureus to grow in the presence of calprotectin. Now, this is fairly indicative that these might be important, but it doesn't really establish that the CNT system or that CNT transporter is actually working with the synthesis and efflux machinery. To test that, we took advantage of the fact that metallophores, or Staphylopine, if it is working as a metallophore, is going to be secreted into the media, and we can harvest that media and rescue strains of Staph aureus that can't make the metallophore by themselves. So I want to take a moment and just walk through this slide here. There's a little bit going on. So here we have strains of bacteria that we're going to use. We have our wild type bacteria. We have a strain that lacks CNTA, so it cannot obtain zinc from the environment, can't transport things. And we have a strain that lacks the synthesis machinery, so it can't make this metallophore. We then provided these same strains with media from either wild type bacteria, that same strain that can't transport the putative metallophore, and media from a bacteria that can't synthesize the metallophore. Here we have whether or not the metallophore is going to be present. So in wild type bacteria, the metallophore will be present. In our CNTA mutant, the wild type, the metallophore will be present. But in the supernatant from the strain that can't make the metallophore, it won't be present. So if we see here, we see that wild type bacteria fed wild type supernatant or supernatant with the metallophore grow just fine. A strain that can't import the putative metallophore doesn't grow. But our strain that can't synthesize the metallophore, if we give it the metallophore and the supernatant, can grow. Similarly, our transporter mutant, or supernatant from our transporter mutant, we see that wild type grows, our transporter mutant strain doesn't grow, and our strain that can't synthesize the metallophore doesn't grow, grows. However, when we give these strains supernatant from the bacteria that can't make the metallophore, wild type bacteria still grow, but our CNT-KLM mutant no longer grows, indicating that these systems, the CNT transporter and the CNT synthesis machinery, are functioning or working together. Now, this tells us once again that these systems are important in vitro or in culture, but they don't tell us that they're important in vivo. So we once again performed an infection experiment. And what we see is that the wild type bacteria, the bacterial burdens we see from wild type are here. If we infect these mice with a CNTA mutant, once again, we see that the CNTA strain has a virulence defect. But if we infect them with a mutant that can't secrete the metallophore, or can't secrete the Staphylopine, we see an even more pronounced virulence defect. Now, so this virulence defect here suggests that we may be able to target this secretion apparatus as a new way to treat staphylococcal infections. It also highlights the importance of being methodical, I think, as we approach science, because one might suspect that a CNTA mutant and a CNTE mutant, because they're working together in the same process, would have similar phenotypes or similar virulence defects. But we see that by asking this question about every component of the system, that some of these components, while they're all contributing to the same process, have larger phenotypes or contribute more to the ability of Staph aureus and perhaps other pathogens to cause infection. So, so far I've told you that Staph aureus uses two transporters to compete for manganese and zinc. We know the CNT system is necessary for Staph aureus to obtain zinc from the host. And we know that Staphylopine is a zincophore, which is necessary for the ability of Staph aureus to cause infection. But if we think about this in a broader context, is this new strategy for obtaining zinc something that's specific to Staph aureus? Do we find something that only Staph aureus does? Or do other bacteria use the same strategy to compete with the host or compete in other environments, perhaps, where zinc might be limiting? So to ask this question, we took advantage of the tools in the IGB and analyzed all of the sequenced genomes of all of the microbes out there and available and asked, Is it possible that any of these other organisms or other microbes could produce Staphylopine-like molecules? And what I'm showing you here is a network analysis where each one of these dots represents a bacteria that contains the machinery necessary to produce a molecule that looks like Staphylopine, looks like that zincophore that's necessary for Staph aureus to cause infection. We can see this group in purple here, this group that contains Staph aureus and the ability to make Staphylopine. This largely consists of other Staphylococci, others which are capable of infecting us, animals, lots of mammals. We see over here in this group that has purple and light blue, a number of bacteria that we can find in the soil. We find bacteria associated with the rhizosphere. If we look over here, we see Yersinia pine, a molecule very similar to Staphylopine that's produced by Yersinia bacteria. This includes Yersinia pestis, the cause of bubonic plague, as well as other Yersinia that cause intestinal illnesses. We look in this group here, we see bacteria that are related to the Pseudomonads. So Pseudomonas aeruginosa, one of the primary pathogens that are found in the lungs of individuals with CF. But also Pseudomonads that live in the soil, live on plants, cause crop disease. We can expand our analysis and look in these groups down here. We see bacteria that grow and thrive in our oceans and waterways, as well as our own intestines. And so by studying Staph aureus, studying a pathogen and asking how does it respond and adapt to zinc limitation, we found a new system that is present in bacteria that grow in the soil, bacteria that grow on us, bacteria that grow on plants, and are likely influencing how these bacteria affect numerous processes we think that are important for humans and our societal benefit. And so just like to leave you with the idea that similar systems to the Staphylopine system are found in a wide variety of bacteria. And so by studying a pathogen, we can gain benefits in terms of treating human disease because the system offers new opportunities to prevent Staph aureus from acquiring zinc during infection. But if we expand our thoughts and think a little more broadly, we perhaps have new opportunities to treat or manipulate the soil to improve crop yield, improve plant health, improve the health of our aquatic waterways with just a little, all because we studied a pathogen. With that, I'd like to end once again by thanking all of the people who did the work, and I'd be happy to take any questions that you have today.
Awesome. Thank you, Thomas. That was great. We're going to get started with our Q&A. Just as I have a couple reminders before we do, though, to ask questions, you can go to c2st.cnf.io. You'll see today's session. You'll click on that and type in your question, and you can see if it gets upvoted, and we'll certainly get to it. As an additional reminder, we are having one of our auxiliary members join us after today's Q&A. So if you're interested in joining or learning more about the work that C2ST's auxiliary board is doing, please hang out after. All right, so to get started, we have a number of great questions in Q&A. So our first one is, on a high level, since typical media for bacterial model organisms such as E. coli is very nutrient rich, how much knowledge do you think we have generalized about bacteria is skewed due to lack of environmental pressure?
So I think that's a good question. I think studying bacteria in rich growth media is extremely beneficial because it gives us an understanding just to ask, how do things work? Many of the fundamental processes that we understand were guided by studies done in bacteria. So how we eat sugars, how our bodies consume sugars was guided by work done in bacteria, guided in E. coli, studies that were done in E. coli in rich medias. So I think rich medias and those things certainly have a place for studying bacteria. And there's tremendous value to using those strategies. I think we have, when we're studying bacteria, we have to remember though that that's not necessarily their natural habitat. So if we're studying and thinking about pathogenesis, we want to acknowledge and then perhaps take advantage of the fact that we can manipulate the metal levels to develop a greater understanding of what's going on.
Great. Our next question is a bit more general. This person asks, What are the differences between a pathogen and a virus? The definitions look pretty similar.
So a virus does not necessarily have to be a pathogen. A virus can be a pathogen, but not necessarily is a pathogen. So a virus can be a pathogen, a bacterium can be a pathogen. A virus is a more refined subset of that group.
Awesome. So we have, I think, along similar lines here, and this is obviously top of mind for a lot of us, if not all of us. Can studying pathogens help us to better understand or combat COVID-19?
So I think that's a great question, and I think the answer is clearly yes. So I'll preface this by acknowledging that I am not a virologist, and that's not my field of study. But the success and rapid rate at which we've been able to begin to come up with, and hopefully which will be successful, treatment strategies and perhaps vaccines for COVID-19 stems from the fact that there was basic fundamental research that's been done by labs around the world over the last decade, studying how these SARS-CoV or SARS-like viruses work and understanding how they replicate, how they infect cells. So just studying pathogenesis writ large and the basic biology of these organisms and viruses really is the fundamental knowledge that allows us to build or create treatments and vaccines for infection.
Interesting. Yeah, it seems like there's a lot of room for collaboration on that. So this next question is, I think, about the last graph that you had in your presentation, and they were asking what the edges of the graph represented.
Sorry,
if you could just give some additional clarification on that.
On the last graph.
Yeah.
Perhaps this one.
I think so. Yeah.
Can you repeat the question then? With that,
they're just asking what the edges in the graph represent.
So these lines here, I think, what they're referring to by the edges, that represents how similar all of these proteins are, how similar these systems are that make the metallophores. Buried within this graph that I didn't talk about today is we can make predictions about what these other molecules produced by these other bacteria look like. And so I call them Staphylopine-like molecules because their core structure we think looks like that of Staphylopine, but the bacteria we think are actually changing them up a little bit, making them chemically look a little different. We don't know why they're doing this, but we think it has to do with their ability perhaps to compete with other bacteria for zinc. Basically, I can use mine, but you can't use mine.
Interesting. And then for this next question, I know you touched on this a bit at the end of your talk, but this person is wondering what practical applications that you hope to see from your research, I guess more specifically.
Yeah, thank you. That's a really great question. So if we think about just the CNT system by itself, there's really several opportunities for therapeutic intervention. So the transporter, it's something that's on the surface of the bacteria. We know, since it's essential for infection, that it has to be made by the bacterium when it's causing disease. So if it's on the surface and it's made by the bacterium, it's potentially something that can be incorporated into a vaccine, and we can train our immune response to target the bacteria. Additionally, we know that Staphylopine has to be made by the bacteria. Perhaps we can come up with compounds or drugs that prevent Staph aureus from making Staphylopine, or prevent Staph aureus from secreting Staphylopine. All of these strategies, I think, would ablate the ability of Staph aureus to cause infection.
Great. And we have one more question, but before I get to that question, we actually have a comment from someone who was watching. They said, please tell Dr. Kehl-Fie that we all care about his son and wouldn't have minded if we'd heard him during this. Great session, very educational. So I think that people overall are pretty excited to see pets and children. I know that my coworkers like it when my dog makes a appearance on these. So for our final question, and this, I guess you can take in whatever direction you like, but what ongoing or future projects are you excited about? And I guess it could be your projects or ones of colleagues, maybe.
So I think there's a lot of really, I'll focus on on just the work I talked about today, I think there's really a lot of exciting opportunities. One we've just briefly touched on is the opportunity for therapeutic interventions to improve our ability to treat and prevent staphylococcal infections, and perhaps infections with other bacteria such as Pseudomonas aeruginosa may improve the lives of people with cystic fibrosis, etc. But also what I'm really excited about is exploring all of these other bacteria that make these metallophores, these zincophores. How do they impact plants? How do they impact microbial communities in the soil? Things like that, because it's really an area of research that hasn't been studied, because until recently we didn't know of their existence. And I think there's really a great opportunity there perhaps to ask other questions and new questions about what's going on and why we have these, or why not we, but the bacteria have these systems.
Well, that I think wraps up Thomas's talk for today. I want to thank him for an excellent presentation, as well as the Carl R. Woese Institute for Genomic Biology and the Kleinman Center for Genomics in Business and Society. I'd also like to remind everybody that we're about to hear from C2ST's auxiliary board, so please stay tuned for that. But also, please go to c2st.cnf.io to evaluate today's program. That was where you were asking questions. So I'd like to bring on Robert Kappers, who is one of our co-presidents of our auxiliary board.
Hi, everybody. Mary, can you see me?
Yeah.
Okay. Hi. So I am a member of the C2ST Auxiliary Board, currently serving as the co-president. And C2ST is such a great organization, and our auxiliary board forms, we're comprised of a number of STEM individuals, engineers, and scientists that provide support for C2ST's staff and the board. And it's a really great organization and a great board. And I'm really here to advocate for others to join us in our mission, and I'm here to answer any questions that anyone may have.
Yeah, I don't think we have too many questions coming in from audience members, but what people can do is reach out to us at info@c2st.org. That's our general staff email that gets checked daily. So if you have questions about the auxiliary board that we can answer, our staff will. We're also happy to pass that along to the appropriate auxiliary board member if you're looking to get in touch with one of them. Okay, we do have a question that just came in, and as a reminder, you can ask these at c2st.cnf.io, the same place you were just asking questions for our talk. Robert, this person is asking, what kind of activities have you been involved with at C2ST?
We do a lot of things on the auxiliary board. So I think some of our primary activities would be to raise funds for C2ST, and facilitate networking for STEM individuals within the Chicagoland area. And so one of the more fun events is our fall social that we're currently planning. But we in a pre-pandemic world, we'll gather at a bar or a brewery and provide for some great networking. And that also provides for a chance to raise some funds for C2ST, and which allows for us to put on these great events.
Awesome. Yeah, and we have another question that's come in, and this person is wondering if you have to be a scientist or strictly affiliated with a science organization to join.
No, I think it's anybody that's passionate about math and science and engineering and technology. My background is in engineering, chemical engineering, but I quickly pivoted from engineering to practice law. And so I'm not really affiliated with a particular scientific organization. I just enjoy math and science, and I'm passionate about C2ST. And so if you're interested, I would welcome you to join regardless of, who you work for, your particular background.
Awesome. Well, thank you, Robert. It doesn't look like we have any more questions. So unless someone squeaks one in here in the last couple of seconds, just as a reminder, you can reach us at any time. That email address is info@c2st.org. You can find that on our website as well. And any more information about C2ST you can find on c2st.org or reach out to our staff. We will get back in touch with you. And if you do have more questions or want to evaluate this program, please go to c2st.cnf.io. Thank you, everybody, for tuning in.